What do I mean when I say something's an activated carrier? These are molecules that carry something called a functional group, which is different for each carrier group pair, that are eventually transferred from one molecule to another during different metabolic reactions. Hint, hint, it'd be a really good idea to know which functional group each activated carrier is associated with. This will help you to determine the output of each metabolic reaction. However, knowing the exact structure is not necessarily important for the USMLE. Adenosine triphosphate, which we just talked about, carries 3 phosphoryl groups. So ATP is the activated carrier, which is carrying the functional group of 1 or 2 phosphates, depending on the reaction. It can use these high-energy phosphate bonds to phosphorylate proteins, which can activate or inhibit the function of a protein, or change how it interacts with other molecules. The process of phosphorylation in a kinase cascade is analogous to a baton being passed in a relay race. For the race, or chemical reaction to proceed, each runner or downstream intermediate has to accept the baton, in other words the phosphate group, from its upstream kinase, and ATP, the upstream runner holding the baton that can be passed on. This analogy breaks down at an important point of distinction. Not all phosphate groups are the same. It's usually just the last of the 3 phosphate groups that's unstable enough to be transferable. NADH, shown here as NAD+, as well as NADPH and FADH2, carry electrons. These electrons can then be traded in later down the line to make ATP. Circled here is where the electrons and hydrogen would go in NADH. Coenzyme A, shown here, and lipoamide, which is lipoic acid conjugated to a protein via an amine bond, carry acyl groups. Did that sound kinda dense? Don't freak out. All you really need to know here, for the test, is that those 2 things, CoA and lipoamide, can carry acyl groups for a bit. What's an acyl group? It's the generic name that we give to hydrocarbon chains. Circled here is where the CoA would bind to the acetyl group. Biotin carries carboxyl groups. Circled here is where the carboxyl group would bind, and tetrahydrofolic acid carries single carbon groups, here. S-adenosyl L-methionine, otherwise known as SAM, carries methyl groups, here. Lastly, thiamine pyrophosphate, which is the active form of vitamin B1, carries aldehyde groups. In the Biochemistry Nutrition section, you talked about the most common electron acceptors, which are the nicotinamides, NAD+, and NADP+, and the flavin adenine dinucleotide, FAD. Thinking back to the Biochemistry Nutrition section, do you remember which vitamin FADH is derived from? FADH is derived from riboflavin, or vitamin B2. On the other hand, NAD+, and NADP+, are derived from niacin, or vitamin B3. All 3 of these will accept electrons using various redox reactions and then transfer those electrons to other molecules, such as the proteins of the electron transport chain. NAD+ is mostly used for catabolic processes that break down organic molecules. And the electrons are carried in reducing equivalents as NADH. If NAD+ is primarily used in catabolic processes, what's NADPH used for? NADPH is used in mostly anabolic reactions, such as steroid and fatty acid synthesis. NADPH can be created using the hexose monophosphate shunt, otherwise known as the HMP shunt, which we'll cover soon. It's also used in other places, like during the respiratory burst, to create oxygen radicals to damage bacteria, in the cytochrome P450 system that we'll cover in pharmacology, and by glutathione reductase to reduce glutathione. Now we'll talk about the first step in glycolysis, which is the metabolic process that breaks down glucose to make ATP. Do you remember what the function of a kinase enzyme is? From our discussion of terminology, the kinases transfer a high-energy phosphate group from ATP to a substrate. We see a cell here and a molecule of glucose outside of the cell. This glucose is free to enter and leave the cell in its current form. Once the glucose enters the cell, it meets with the enzyme hexokinase, or glucokinase, which adds a phosphate group to the glucose molecule. Thus, the first step of glycolysis is phosphorylation of glucose to create glucose 6-phosphate by using 1 ATP. Why is a phosphate added to the glucose? In addition to preparing glucose for the next steps in glycolysis, this also gives glucose a negative charge, which prevents it from passing through the plasma membrane, effectively trapping the glucose in the cell. This reaction can be catalyzed by either hexokinase or glucokinase. The differences between these two are their locations inside the body and enzyme kinetics. We'll talk more about enzyme kinetics in pharmacology, especially Km and Vmax, which you see here in this graph. For now, however, I'm going to focus on the kinetics of these two specific enzymes, hexokinase and glucokinase. Hexokinase is found in almost every cell in the body, except for hepatocytes and pancreatic beta cells. Hexokinase has a high affinity for glucose, but has a low maximum reaction rate. This means that the concentration of glucose needed for this enzyme to be used is relatively low, since it has a high affinity. But the speed at which the reaction occurs is also pretty low due to its low Vmax. Hexokinase is not affected by insulin, but does receive negative feedback from high amounts of glucose 6-phosphate. So basically, when the cell has low glucose 6-phosphate, hexokinase will catalyze any glucose available to glucose 6-phosphate at a low rate. However, if there's a large amount of glucose in the blood, such as after a meal, hexokinase won't be able to keep up since it has low capacity. What do you think will be the effect of all that extra untrapped glucose floating around in the context of an otherwise healthy person? What about in the context of insulin insensitivity seen in diabetes mellitus? You'll see more on that in the endocrine chapter. Glucokinase comes into play after meals, when there are high glucose loads. Where is glucokinase found in the body? Glucokinase is found only in the liver and in pancreatic beta cells. This enzyme has a low affinity for glucose, but a high capacity or reaction rate. This means that the glucokinase is not going to be active unless you have a lot of glucose. But if you have a lot of glucose, it can act very quickly to phosphorylate all of it. Also, it has no feedback inhibition from glucose 6-phosphate. As long as there's lots of glucose in the bloodstream, glucokinase will keep phosphorylating glucose inside of cells. You could think of this as being kind of like a buffer system. Because after you eat a large meal, glucose will activate beta cells in the pancreas, which will then release insulin, further activating glucokinase in the liver so that the glucose is removed from the blood and stored as glycogen. If it helps, you may remember that glucokinase is found in the liver since both liver and gluco prefix have 5 letters. What's another way to think of hexokinase and glucokinase? I like to think of these two enzymes as a tortoise and a hare. Hexokinase is the tortoise because it acts at a slow and steady pace. Glucokinase is kind of like a hare. I like to call him a lucky rabbit, speeding up and slowing down depending on the amount of glucose in the blood. I also want to touch upon the concept of maturity-onset diabetes of the young, or MODY for short. Again, you'll learn more about the disease diabetes as a whole in the endocrine section, but in a nutshell, diabetes results in an increase in glucose in the bloodstream. There are many different forms of diabetes. Maturity-onset diabetes of the young is a group of monogenetic diseases, meaning there's usually only one mutation in one of a few genes causing the diabetes, versus other forms of diabetes which usually have multiple genetic or non-genetic causes. One of the mutations in MODY can involve the glucokinase gene. This mutation will functionally alter the glucokinase enzyme, producing a form of diabetes. Why is this? Remember that glucokinase is induced by insulin when there are high levels of glucose in the bloodstream. If a point mutation causes the glucokinase enzyme to malfunction, insulin will not activate glucokinase like it normally would, resulting in increased levels of glucose in the bloodstream because glucose would no longer be trapped inside of cells. It's important to note that hexokinase has not been associated with MODY. Okay, flash quiz time. Let's review. A 65-year-old with type 2 diabetes injects insulin before his lunch. How does insulin affect the action of hexokinase and glucokinase? I'll give you a second. Hopefully, you remember that insulin induces the enzyme glucokinase, but doesn't affect hexokinase. This is because hexokinase is always active, while glucokinase must be stimulated by insulin when glucose levels are high to create more glucose 6-phosphate to trap glucose inside of the cell. Shown here, we see the glycolysis substrates, intermediates, and end products. I'll focus on the important steps, but I wanted to first offer a global picture of glycolysis. Notice how most of these steps have double-headed arrows, meaning that they can occur in both the forward and backward directions. Let's focus on the unidirectional arrows. Here, we see the 3 steps of glycolysis that are irreversible, meaning they only occur in the forward direction. The net reaction of glycolysis is shown by this equation. You start with a molecule of glucose, 2 inorganic phosphates, 2 ADP, as well as 2 NAD+, and end up with 2 molecules of pyruvate, 2 ATP molecules, 2 NADH and hydrogen ions, and water. What's important here? You need 1 glucose to make 2 pyruvates and 2 ATP, and in the process, 2 NADH plus molecules get reduced to form 2 NADHs and hydrogen ions. Lastly, water is generated as oxygen accepts electrons at the end of the electron transport chain. If you get confused by which species gets reduced or oxidized, don't freak out. Look at the charge. Here, NAD+ changes to NADH. NAD+ loses its positive charge. In other words, its charge is reduced from +1 to 0, or neutral. Now, let's go through the enzymatic steps that regulate glycolysis. The 2 steps in glycolysis that require ATP are the first and third steps. This will represent our initial energy investment phase. First, we said that hexokinase or glucokinase uses 1 ATP to convert glucose to glucose 6-phosphate. As I mentioned before, glucose 6-phosphate exerts its negative feedback on hexokinase, but not on glucokinase. Instead, high levels of fructose 6-phosphate wind up inhibiting glucokinase. In the third step of glycolysis, phosphofructokinase 1 uses 1 ATP to convert fructose 6-phosphate to fructose 1,6-bisphosphate. This just means that it's fructose with phosphate groups on the 1 and 6 position. Do you remember what the rate-limiting enzyme was for glycolysis? Phosphofructokinase is the rate-limiting enzyme in glycolysis, so there are a lot of things that can either activate or inhibit it. If cells have lots of adenosine monophosphate, or AMP, this means that they need to make more ATP, so PFK1 will be activated. If there's a lot of fructose 2,6-bisphosphate, this indicates that blood sugar is high, so there's plenty of glucose to use for more glycolysis. Therefore, both of these activate phosphofructokinase. If lots of ATP and citrate are present, this means that the cell has a sufficient amount of energy available and does not need to continue glycolysis. There are 2 steps of glycolysis that produce ATP as well, but both of these occur twice, since they occur after the step when our 6-carbon molecule is split into 2 3-carbon molecules. This means that a total of 2 ATP are initially consumed, and a total of 4 ATP are produced per molecule of glucose, resulting in a net gain of 2 ATP, which I mentioned in the overall glycolysis reaction previously. We've already invested some ATP prepping this glucose molecule. So when do we finally make some ATP? The first step of glycolysis that produces ATP uses the enzyme phosphoglycerate kinase. Phosphoglycerate kinase converts 1,3-phosphoglycerate into 2-phosphoglycerate. Going by the names of the molecules, it makes sense that 1 ATP was produced, since 1,3-bisphosphate has 2 phosphate groups, which are at the 1 and 3 positions, while 3-phosphoglycerate only has 1 phosphate group. So that means one of those phosphate groups was taken off to make ATP from ADP. Why does the enzyme phosphoglycerate kinase work twice as hard as hexokinase and phosphofructokinase 1? This enzyme has to work twice as much as hexokinase and phosphofructokinase 1 because halfway through glycolysis, the glucose molecule is split in 2. These 2 smaller fragments, called glyceraldehyde 3-phosphate, undergo the same reaction to form 2 molecules of pyruvate. Finally, pyruvate kinase converts phosphoenolpyruvate into pyruvate, producing 1 ATP in the process. This is activated by fructose 1,6-bisphosphate, which is an intermediate of glycolysis, and inhibited by high amounts of ATP and alanine, since again, if you already have a lot of ATP, you don't want to keep going through glycolysis to make more. To repeat, this enzyme works double time in glycolysis, just like we saw before with phosphoglycerate kinase. And that's it for the glycolysis pathway. On to the next metabolic process, the TCA cycle, otherwise known as the Krebs cycle. Pyruvate dehydrogenase is the enzyme that links glycolysis, which takes place in the cytosol, to the TCA cycle in the mitochondria. Okay, flash quiz time. In glycolysis, which reactions require ATP? I'll give you a little bit. There are 2 steps in glycolysis that require an investment of ATP: converting glucose to glucose 6-phosphate and fructose 6-phosphate to fructose 1,6-bisphosphate. It's like investing on Wall Street. You need to initially invest some money, or 2 ATP, before you get any return on your investment, or 4 ATP, and some NADH and hydrogen ion. We already took a look at glycolysis and in part how it's regulated. It's pretty logical to understand why end products such as ATP can inhibit glycolysis. But the reason why fructose 2,6-bisphosphate can activate some glycolysis enzymes may not necessarily be as intuitive. How does fructose 2,6-bisphosphate fit into the glycolysis pathway? Think of fructose 2,6-bisphosphate as a regulation molecule or a side process that's going on along with glycolysis. It gets made by phosphofructokinase 2, a kind of cousin to phosphofructokinase 1, and gets broken down by fructose bisphosphate 2. Fructose bisphosphate 2 and phosphofructokinase 2 are actually part of the same protein, but respond in opposite ways to phosphorylation by protein kinase A. Let's compare the regulation in this enzyme in the fed and fasting state. In the fasting state, high glucagon increases the amount of cyclic AMP through a G protein-coupled receptor system. This increases the activity of pKa, which phosphorylates fructose bisphosphate 2. This phosphate group activates fructose bisphosphate 2 and inhibits phosphofructokinase 2. This means you're going to be converting this side molecule of fructose 2,6-bisphosphate into fructose 6-phosphate and inhibiting the opposite reaction, which in turn allows gluconeogenesis to proceed to replenish blood glucose levels, which makes sense if you're in the fasting state. What do you think will happen in the fed state? In the fed state, high insulin levels decrease the amount of cyclic AMP, and therefore decrease protein kinase A activity. The enzyme fructose bisphosphate 2 will be dephosphorylated, which means that only phosphofructokinase 2 will be active. The body will start to convert fructose 6-phosphate into fructose 2,6-bisphosphate, which activates the forward reaction by phosphofructokinase 1, which allows glycolysis to proceed. Just remember, the molecule fructose 2,6-bisphosphate is made to regulate the steps of glycolysis. Keep in mind that phosphofructokinase 1 is the rate-limiting step in glycolysis, and therefore it's important to regulate this enzyme more than the others. Okay, flash quiz time. A hardworking medical student on her surgery clerkship skips lunch to go to the OR. How does this affect the action of fructose bisphosphatase 2 and phosphofructokinase 2? I'll give you a bit. In the fasting state, increased glucagon levels increase cyclic AMP levels and protein kinase A activity, which phosphorylate fructose bisphosphatase 2, ultimately activating it, which lowers glycolysis and increases gluconeogenesis.